I think most people, when they think of DNA, if you have any idea about iconic scientific images, one of them is B-form double helix, that twisted ladder sort of structure, which is I think it's an iconic scientific image of the 20th century. So this is what Watson and Crick first proposed as a structure for DNA in 1953. But we've known for a very long time that DNA can do lots of weird things. So even in the early 1900s, if you dissolved guanine or guanosine, which is one of the individual bases that makes up DNA, if you dissolve that, you get a gel. And that shows that that particular nucleotide has forms of self-assembly. And that was a long time before people were even really interested in the structure of DNA. And then in the 1960s, a lot of work was done to look at how the different bases can interact with each other. And a scientist called Hoogstein was investigating that, and they found that actually you didn't have to have these matching pairs that Watson and Crick first proposed. They didn't have to match. They could do other things.
Welcome to Demystifying Science, where we're exploring the edge of what is known. Today on the show, we have Dr. Zoe Waller, who is doing all kinds of really interesting work concerning the alternative structures of DNA. Normally, DNA is thought of as this double helix, the Watson and Crick base pairing. But that paradigm is expanding, and Dr. Waller studies some aspects of that, and she is knowledgeable about many others. So could you go ahead and sort of orient us to what the problems that you're working on in your lab and how you got interested in that, where you're coming from? What's going on? Dr. Waller, welcome.
Thank you for having me, and especially to talk about special types of DNA structure. It's my favourite subject, so thank you very much for the opportunity. So I think most people, when they think of DNA, if you have any idea about iconic scientific images, one of them is B-form double helix, that twisted ladder sort of structure, which is I think it's an iconic scientific image of the 20th century. So this is what Watson and Crick first proposed as a structure for DNA in 1953. But we've known for a very long time that DNA can do lots of weird things. So even in the early 1900s, if you dissolved guanine or guanosine, which is one of the individual bases that makes up DNA, if you dissolve that, you get a gel. And that shows that that particular nucleotide has forms of self-assembly. And then also in the 1960s.
Is that because the gel has like a matrix to it? Is that what you mean?
It's because guanine is really unique it's very special in the sense that it's very self-complementary. So in normal double helical DNA, guanine likes to base pair with cytosine. So you have G and C base pairing. So there are 4 nucleotides, A, T, C, and G and C love each other. But what we know is that G also loves itself. So
it makes a kind of lattice when it's in solution, this gel that you're referring to.
So it doesn't make sort of a lattice. So guanine, I'd like to think of it like a corner. So it has 2 what we call hydrogen bond donors on one side and 2 hydrogen bond acceptors on another side, and they're a perfect complement. So it can form little squares. So they're like corners, but it can also form helices. And this is where the gel formation can happen. So you can have squares of guanines or tetrads that stack on top of each other, or you can have these kind of helical molecules that get entangled and they can form a gel.
What is a gel, by the way, just for everybody out there?
A gel, I think most people are probably familiar with hair gel. So it's like a semi-solid. So it's kind of hydrogels have a lot of water component, but it's not quite a solid. It's not quite a liquid.
And the rigidity component comes from these repeat units?
Yes. So something's holding that together. So it's not just a complete fluid like a solution would normally be.
Got it. So I didn't mean to derail you. I'm just trying to understand.
It's fine.
So this is the first sort of hint that maybe these bases are doing something structural or something other than simply serving as this matched code sequence.
Absolutely. And that was a long time before people were even really interested in the structure of DNA. So they were understanding about different components of it. And then in the 1960s, a lot of work was done to look at how the different bases can interact with each other. And a scientist called Hoogstein was investigating that. And they found that actually you didn't have to have these matching pairs that Watson and Crick first proposed. They didn't have to match. They could do other things. And that the types of interactions between those bases are now termed Hoogstein hydrogen bonding. So Watson and Crick hydrogen bonding is what you see in a normal double helix. And then Hoogstein hydrogen bonding is basically anything else.
So is this the same base pairs interacting in a new way, or is it different base pairs interacting?
So each of the bases that DNA is composed of, so there's 4, each of them has the capacity to pair up with their natural partner, but there are different elements of their structure that enable them to do other things. So with guanine, it has a face, part of that molecule has a face that will interact with cytosine, but it has extra interactions on another face of that molecule where it can do other things.
Got it.
And it's these extra additional capabilities that these bases can do, which then extend the structures that they can form.
And so when we talk about Hoogstein base pairing, so Watson and Crick is ATCG, but for Hoogstein pairing, you said that it doesn't necessarily have to be with a complementary molecule. So would the tetrad formation of guanine complexes be an example of Hoogstein base pairing?
Absolutely. Yeah, so that's exactly right. So anything that isn't Watson and Crick is largely described as Hoogstein base pairing. So it's not like there's any specific types of Hoogstein base pairing, although the original ones were kind of the classics, but it's anything that isn't Watson and Crick is generally described as Hoogstein, sort of alternative base pairing.
And was
alternative interactions.
Excuse me, was it immediately apparent to folks when they discovered that these pairing possibilities were out there? Was it immediately apparent that they probably served a function, or where are we at right now with the functional aspect of these alternative structures?
Well, there's quite a big gap between finding out that the bases can do some other types of hydrogen bonding or interactions. That was the first step towards maybe they do something different. And then in 1962, that was the first time they showed that guanine can form these squares, these tetrads that stack on top of each other. And then other structures were shown. For example, in the 90s, it was shown that cytosine could also base pair with itself. And you can have CC base pairs. And there have been lots of other structures shown since, but largely we're now looking at, well, we know that these structures now sort of exist. There's plenty of evidence that they might have a biological function, and now actually what potentially do they do? And initial studies were focused on looking at cancer and how they might be affecting in cancer. Because of different aspects of looking at where they are in the human genome. And So, for example, some scientists at Cambridge that were led by Shankar Balasubramanian, and one of the key players in that was Julian Huppert, used all of the information from the human genome and their understanding of which sequences could fold into special four-stranded structures called G quadruplexes. They correlated what they thought, right? Okay, so we know this type of sequence can form this type of structure. Where are they in the human genome? And what does that mean? And they found that the closer you get to a transcription start site, so that's the point at which a gene starts to be read. So this is a very important part in the genome, which governs, is this gene switched on or not? The closer you get to the transcription start site, the more likely you are to find a sequence that can fold into a G quadruplex. So one of these alternative structures.
And has there been work done to sort of look to see how the G quadruplexes change over time? Because I can imagine that if you have a lot of topology at the transcription start site, it could be a case where it prevents the transcription factor from being able to bind in order to actually transcribe the gene. Or on the other hand, it could act sort of as something that emerges out of an unfolding of the strand in order to actually provide a docking site. But I will say that generally when I've seen transcription factors bound to DNA, the DNA seems to be helical inside the pocket.
Yeah, I mean, I guess that's, if that's what you're looking for, that's what you're going to find, right?
Yeah, that's definitely true.
And I do think that these structures are probably dynamic.
Mm-hmm.
So they're not going to be always in one form.
Because they're affected by temperature and pH pretty significantly.
Yes, so these structures will be affected by a large number of environmental conditions, and the G quadruplexes in particular are very strong and very stable under physiological conditions. And these can be unwound by particular native or naturally occurring enzymes and proteins. So helicases unfold these structures, and that's actually well accepted and well known. So it does show that biology has a way of dealing with these. So they provide a roadblock essentially. But you're right that they could, if there might be an equilibrium between the double helix and one of these structures, and it might be that the transcription factor that we've seen crystallized with the B-form DNA, if they had looked at the single-stranded quadruplex or eye motif or other structure, they may well find that actually that binds that one better.
Can we sort of draw a picture of what these look like for the listeners? What is a quadruplex or the eye motif or something like this? Maybe this would allow folks to sort of understand what we're talking about here.
So a G quadruplex I would best describe as a cube-like structure. So to form that, you have guanines that Hoogsteen hydrogen bond with themselves, and they form squares.
And it's kind of like planar square, right?
Yeah, planar squares that stack on top of each other. So I think one of my students described as stacked square plates. And I was like, oh, that's quite cute. But within the core of the structure, there's like a capacity to bind cations, so small metal ions like potassium or sodium. And so that's the kind of general structure of the quadruplex. There are loops of nucleotides that connect these corners. So
what's up with those metal ions? What's going on there? I just kind of jumped.
Why are they interested? Why are they hanging around?
Yeah. It looks a lot like it kind of looked when I was looking at the picture, it kind of reminded me of like a heme group or something strange like that. I mean, I know it has, it just from like looking at it, I know it has nothing chemically similar, but the metal being stuck there just seems really electrically interesting.
And some people have used these in combination with heme to actually, quadruplexes have been used as, how best to put it, as sort of catalysts for reactions and things like that, and redox potential. But you're right, actually, heme does have, structurally a lot of similarities as such, because it's planar and then it has a metal in the middle. So the metals are there because when the guanines are all in their square formation, they have an oxygen as part of their structure that's kind of all pointing inwards. And oxygen is what we call electronegative. So there's a lot of electronegativity kind of in that core. So cations like potassium, which are positively charged, are quite attracted to that. So some cations sit in plane with the tetrad or the square, and some sit between the plates.
And this is an unfolded. This forms when the sort of normal helix of the DNA has kind of opened slightly in order to create this sort of this. It's a very strange looking shape. When you look, and we can put on the
like
screen.
A wrinkle or something.
Yeah, it's like almost like a wrinkle, like it's opened and then it's kind of folded up onto itself in order to create this kind of this bump on the surface.
Mm-hmm. Yeah.
And the eye motifs are very similar.
So in some ways similar, in some ways not. So they're four-stranded. So quadruplexes, by virtue, you can tell by their name, quadruplex means four-stranded. So the G quadruplexes are composed of 4 strands. So it's like one strand kind of folding back over itself a couple of times to get those interactions. Eye motifs are very similar in the sense that they are, again, one strand that can fold back upon itself. But rather than having squares stacked up on top of each other, the way that this structure is held together is by intercalated base pairs. And the best way to kind of describe that is maybe if you imagine your base pairs being your fingers and you interlock them, that crisscross kind of core of that structure is very tight, very, very different to like a twisted ladder, like the double helix, but also, again, very different to that stacked planar tetrad core of the G quadruplex. So they do look very different.
Back to the function of these structures. Have people been proposing? You mentioned could be catalytic activity with these. Would you call that magnesium intercalated also? I'm not sure how you. But with this structure in general, what sort of processes? I know you're talking about it perhaps regulating binding, of transcription factors, which could be interesting for cancer or developmental biology. What else is going on? What else has been proposed?
So yeah, as you're right, that people have proposed that they may play a role in transcription. And as I mentioned, that you're more likely to find one the closer you get to transcription factor binding site. But depending on how you estimate whether something's likely to form one of these structures or not, the first study showed or indicated that 43% of all genes contained one of these sequences in their promoter region. But 69% of oncogenes, so cancer-related genes, contain one of these structures in their promoter. And so there seems to be this relationship between formation of these structures and either cancer development or regulation of cancer. So people are looking into that. These structures also form at the telomeres, so the regions of DNA that cap the chromosomes. And there is actually an enzyme called telomerase that can elongate the telomeres. And most of the time in normal cells, this enzyme is not really active. It's very active in stem cells, but telomerase elongates the telomeres and essentially kind of makes those cells live longer than they normally would. But 85% of cancers upregulate this enzyme and continue to elongate the telomeres, so that the cells have no idea how old they are, and the older they are, the more mistakes they make, etc., which was bad, and that's one of the reasons why cancers develop. So there's this relationship, because these structures form in the telomeres, that they can actually stop that process. They can actually stop telomerase from working. So that's another area that people have been looking at
naturally, like sort of like natively, as a sort of anti-cancer mechanism or something inside of the cells. Or
yes, people have been looking at that,
or potentially as a therapy as well. Or is there ways to induce these structures? Is that on the horizon?
So yeah, there's a lot of compounds that have been found to stabilize these structures, and some of these have been developed for clinical benefit. Some of them have gone into clinical trials. I think there are some that actually, I can't think of any that have made it into the clinic as yet, but there are many that are currently in clinical trials that look promising. I think sometimes the challenge with working with these structures is because most people are focused on traditional chemotherapeutic, that knowing what we should be doing with these structures and what types of cancers, for example, we might want to be targeting, might be a bit more difficult to work out and understand, and we need to do a lot of learning to that to know what we're effectively trying to do. I say we, like it's not a particular area that I'm like
we need to know what's broken before we try to fix it, something like that. Or we need
absolutely.
It's generally a good approach.
And so there's those but other conditions. So work in my laboratory is funded by Diabetes UK, which is a UK charity which focuses on diabetes, and we are looking at potentially how we could switch on insulin gene expression by targeting alternative DNA structures, as well as also potentially being able to look at the sequence of different people and be able to kind of understand what type of structures that might form, and does that have a role in their potential to develop diabetes.
And what have you found in that direction so far? That sounds very interesting.
Well, we have quite a lot of compounds that switch on insulin, which is cool. And I think working with beta cells is quite difficult. So human beta cells, they're very difficult to propagate or grow. So if you there aren't any standard cell lines that you can use. So we use a mixture of rodent cell lines, and then we're going to use some special fancy kind of created cell line to look at what we're doing. But these compounds work in cells that shouldn't normally produce insulin. So that's pretty weird.
Is there any luck with putting that into animals or clinical significance? Or?
So we are currently focused on working out what on earth these do and how they work.
Yeah, because that's kind of an astounding result, to discover that a cell that's not supposed to be producing insulin, when exposed to a sort of a shape-changing chemical, then starts to produce insulin. That seems like that in itself is plenty to mine in terms of mechanism and understanding.
It seems like somebody would want to market that.
You're such a technologist.
Well, yeah, but I mean, at the same time, you kind of, that's cool, but do you want all of, if you were to give this as a drug, I'm playing devil's advocate here.
Please, yeah.
Do you want all your cells to produce insulin at the same time? So it's kind of good and it's kind of cool, but also
that's a one-way trip. It seems like, yeah.
Is that what we want? Insulin everywhere. So we've got to learn more about it. That's the key thing.
Go ahead.
This seems like a very, very difficult topic to study, because you basically have something that is shape-based, but shape-based on an incredibly small scale. And it's something that may or may not be transient. And so what are the biggest difficulties and biggest successes as well in being able to look into something that is so small and so physical, right? Because transcription is an expression downstream of, okay, you can measure the RNA, you can measure the protein. There's all of these different, like very well established mechanisms for being able to look at transcription and translation. But shape of the DNA.
Yeah, like what tools do you use to even know that this is happening? Have you done AFM, or how does this work? How do you see it?
How do you see it? Yeah, so we use a lot of spectroscopic methods to determine the type of structure that DNA can form in. So we actually use some simple methods like UV spectroscopy. So we can use only just a UV spectrometer and look at the absorbance of the DNA in the folded state and the unfolded state. And if you subtract one from the other, that gives you a characteristic spectrum that you can use for different types of DNA. So I can tell a difference between a quadruplex, an I motif, B form, triplex, like other weird structures, hairpins. They all give like a different type of characteristic signature.
And that's basically just shining a light on it and seeing, what comes, the shadow, I guess, if you will.
The absorbance when it's folded and the absorbance when it's unfolded will give you a different spectrum. Absolutely. So that's a very simple method that we use to determine what type of structure we've got. My next method of choice in my lab is called circular dichroism. So this is it's an interesting method.
That might be the most scientific word we've had on the show so far.
Yeah.
Well, can you say it one more time, actually? What is it?
Circular dichroism.
Circular dichroism. All right.
So it uses circularly polarized light, which is it's kind of a weird concept, but essentially molecules that are chiral. So that means that they can rotate the plane of light, but they also have some sort of asymmetry to them. So chirality is sort of inbuilt into DNA because you've got a right-handed double helix. It's always going to have different shapes around it. And because of the different ways the DNA can fold, we can then monitor this three-dimensional kind of change using CD or circular dichroism. So with the quadruplexes, because they're composed of 4 strands, some of them, if they're all those strands are running in the same direction, we would call that parallel. And if they are running in different directions, we would call that anti-parallel. But we can tell the difference between those using circular dichroism. So again, we can tell the difference between different types of DNA structures, but then also
even the sort of like the interior organization of the structure, because you said that what you're suggesting is that there can be like multiple different organizations of the strands inside of something like a G quadruplex.
Yeah, so we can tell the difference between a G quadruplex and an I motif, but we can also tell the difference between different types of G quadruplexes using that technique.
And are these sort of...
We can't see it, but we can get information about it.
Has anyone actually sort of, because I mean, I've seen some studies that I think that there was something about I motifs, there being an antibody that's actually capable of binding specifically to the shape. And so like antibodies are the sort of the in vivo shape-based assay of choice, I feel like.
Mm-hmm. Yeah.
But has there been imaging? Like, is anybody trying to take, like Dr. DeLay mentioned, AFM or something that actually you have the structure and you're actually sort of trying to image it, or is that too difficult?
Yep.
SEM even? I don't know if people do that or...
Yeah, so people have used AFM to visualize the structure. Like you can see it as like blobs.
Yeah.
So, and what you can then do is you can produce conditions that you know you're going to form these structures in, and you discover those conditions using the techniques that I've just described, UV and CD, nice and cheap and easy. And then you go do your proper experiments using AFM or something similar, and you use those conditions and you go say, well, for example, I motifs. I know for this sequence, if I take the pH really low make it nice and acidic, we're going to get more structures form. And then you'd be able to see that actually in the AFM.
Mm-hmm.
How tightly?
But you're also right in the sense that.
Oh, go ahead. Go ahead. I have a question, but go ahead.
I was going to say, but the antibodies have also been used as well. So some work that I was involved with last year got published last year, and the principal investigator was Nicole Smith, who's based in Australia. She used, so we did some work to show a compound that can help stabilize eye motif structure. And then she used each of the antibodies, one that visualizes eye motifs and an antibody that visualizes G quadruplexes, and in cells showed that if you stabilize the quadruplex, you destabilize the eye motif. And if you stabilize the eye motif, you destabilize the quadruplex by looking at the number of foci or individual kind of lit up bits in the cell visualized using the antibodies. So that's kind of another way of visualizing them. But we can see them using AFM. We have crystal structures of some of these DNA structures as well. So that's another method we use.
Do you think that there are others besides the quadruplex and the eye motif that are waiting out there to be seen, or?
Oh, there are already loads of structures that have already been discovered. So you can get junctions of DNA, and these have been known for years, like Holiday junctions, named after Robin Holiday. There's quite a lot of these things are, named after the scientists that discovered them. So that junction DNA is like two double helices that kind of crossed over almost. You kind of meet kind of almost like a crossroads. You can also get triplex DNA. So this is where you can have another strand that sits in the major groove. So in the double helix, there's a major groove and a minor groove. So one bit's kind of a bit bigger than the other. And that's how you get that kind of quite snazzy asymmetric helix. And it's possible to fit in another strand of DNA in the major groove, and that will give you a triplex. So that's another structure. And
it almost seems like an overwhelming degree of complexity that's emerging concerning this entire transcriptional process. And it's almost like there was a nice happy moment, I don't know, in the early 2000s or maybe the late 90s, when everyone was like, well, it's genes make proteins and this should be easy. We'll have it all figured out. We're going to sequence the human genome and, we'll basically be able to cure every disease in the next 10 years. And now it's almost like the closer we look, and we see this in science in general, anywhere we look in nature, it just gets more and more complex the closer you look at it. I just find it fascinating. I don't know if that was a question. I just am
I suppose I mean, it is amazing. And I suppose my question from that is, what percentage of the DNA is featured like this? Right? Because, you look at, I think that Hoogsteen base pairing is reported to be something like 1% of the DNA. Ish? Is that accurate?
It depends on what the conditions are.
I
It
see.
really is going to depend very much.
And how healthy the patient is. And
yeah. But I think one of the things that came out of the Human Genome Project is, the biggest fact that I think is fascinating is the fact we've got 3 billion bases, and only 1% actually codes for protein. And then they used to think, oh, well, all of the rest, this junk DNA. They called it junk DNA in the 90s.
That's right.
And you can even, I think there's even reference to it on the X-Files and things like that, sci-fi at the time, because it was quite exciting that we might have DNA that actually is not used, and that perhaps it's a bit like our appendix, that once upon a time it was useful, but it's just basically, I don't know, a genomic fossil that we
just a leftover from how fast we evolved,
Yeah, but it soon turned out that if you delete these bits of junk DNA, that you see disastrous consequences, and that's where the whole aspect about learning about non-coding DNA. So the DNA that doesn't code for protein, but is essential to determine how that protein is essentially read. So the DNA read to make the protein. So then if you've got a lot of DNA that's non-coding, that's a lot of information.
It's like an instructional content.
Yeah, like metadata almost. But then how that gets read, that can be read in many different ways. And there's a whole sort of field that's emerged out of how your genetics or how your genome gets read and how that varies, but not in terms of how it changes in the sequence, but how your DNA is bookmarked, like with methylation of cytosine, or how open or closed the DNA is wrapped around proteins. And all of these play big roles in whether genes are switched on or off. And the number of ways that ways there are to kind of exploit that seem to be ever expanding.
What do you think is, so when you look out onto the world of science and you have this, you're studying something that you realize is kind of at the cusp of something very powerful, right? This shape-based functionality inside the genome. Do you ever look out onto the world of science and see other things that you're like, I don't think we're right about this? The same way that someone could have looked at the idea of junk DNA and said the same thing?
Yeah, there are certain things that I look at and I'm like, no, I'm not sure about that. But then when you don't know the ins and outs of a particular field, it's very easy to look and go, nah, not sure about that. But
true.
Only when you kind of feel like you've got a good understanding. And I feel like I've got a relatively good understanding of the DNA structure field. And I'm like, pretty sure there's something more to this. If somebody said, oh, actually, B-form double helix, it just isn't actually relevant. Be like, okay, yeah, I'm on board with that. I'm not surprised, actually. And if there was evidence to show that transcription factors don't bind double helical DNA, they actually bind a weird bulge in Hoogsteen that's Hoogsteen hydrogen bonded, and that actually the crystal structures that we've seen aren't what is actually dynamically relevant and biologically relevant. But the problem is when you're looking at things that are small, it's difficult. Well, you can't see them. So it's about looking at the evidence that you have at the time and making the best judgment of what you can, the best hypothesis.
Yeah.
And especially inside of the cell, it seems like you have various indirect ways of visualizing these, but it seems like one of the most difficult things in the world is seeing it in its native context, which might mean everything in terms of the chemistry of the cell. And one thing that you mentioned I wanted to, before we moved past this too far, was that the pH can affect whether these structures form or fold or unfold. Mm-hmm. How variable is the pH inside of different cells at any given moment? Is this something that swings wildly from individual to individual or time of day? Or I genuinely don't know, and I'm curious.
So this is a question that I don't think's actually been well resolved. So there are schools of thought that state that cells are maintained at pH 7.4-ish, and there are some reports that cancer cells are more acidic. Mm-hmm. But there are also some reports that say that the extracellular matrix is more acidic, but the inside of the cells is not. And people aren't really in agreement on it. So I do think it's an open question.
Okay.
And even if generally the overall internal compartments of the cell, you could say, oh yes, well, it is pH 7.4. You don't know what the localized pH of any single point is,
Right.
really. And people are developing probes to be able to test that. But even that, they've got to be small to understand what the localized pH is going to be.
What about NMR or something that's where you? I'm trying to think of a way that we could look at that.
Well, I know that there's been some, if you so let's take a step back. Do you think that you can use the redox state of the cell as a proxy for pH or no?
In some respects, I guess that's what people do for certain cell types. But again, individual parts of the cell are going to have localized environments that are going to be, I think, very different. And I think that's why people can measure things, but that doesn't necessarily mean it's representative of what is there. And so that's why I think there's still a debate.
Yeah, and I mean, there's some movement towards creating these sorts of small molecule redox probes where you put them inside the cell and they're able to change their fluorescence based off of the conditions within the cell. But everything that I've seen so far, like we tried to use them in my lab when I was in grad school, and they were very finicky. They were very prone to sort of incomprehensible results that didn't really give you what you were looking for. And I think that that's kind of the problem when we start talking about, well, what is happening inside of a cell? Because the methods that we have are generally methods that are good at taking bulk quantities of something and being able to say that, like, well, look, we have some amount of stuff and we can tell you something about it. But when you start to get so small as to the order of like a specific area of the cell, you kind of start to lose the thread of, for me, I think even sort of techniques that you could imagine being functional enough to report something back to you.
Yeah, I would agree. And actually, you might have a probe that works well in dilute solution and, under conditions that you can control. But in the cell, it's very crowded molecularly. There's a lot of stuff there. And what might be really useful in a dilute solution may not work in the same way in the cell, because it's very crowded, and it might bind with a protein and do something else that's weird, or and you can't test against every protein or every lipid or other biological molecule that it could potentially encounter. And some of these probes, they like certain parts of the cell more than others, because if your probe is very hydrophobic or lipophilic, it's going to go to places where it's lipophilic. The cell's also going to then make modifications to your probe. As soon as something goes in, that cell's going to be like, what is this?
Yeah, exactly.
What is this? Don't think this should be here. And then things start getting metabolized and things like that. So, which is natural. So we've got to bear these things in mind when we're making these types of probes. And that's why whenever you're looking at a problem, it's always good to look at it using different things, different methods. And then you can say, okay, well, none of these methods individually is telling me something sort of particularly noteworthy. But actually, as we gradually build up the picture around, this makes it more clear. But you're right, the pH of the cells is a very interesting and open question. It's not one that I started reading the literature about this and I was like, oh, I can't really agree. So I'm going to just not touch that. Because I think sometimes people would say, oh, well, we're working on eye motifs because blah, 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 blah. People have said this. And I look at that and I'm like, a lot of people say the opposite. So
and that's
you could say that about any science, but I just think you just need to be careful about making statements, because sometimes statements that aren't accurate can be propagated in the literature. And actually, if you then go back to the original paper, I'm like, they didn't say that at all, actually. You're just wishful thinking.
We've seen this happen a few times. It brings up this really interesting moment that we're at in science right now, where there's so much information, and finding a systematic way of pulling patterns out of that greater sea of information is really going to be an interesting hurdle for the next generation.
Because the conflicting information might not be that one person is right and the other person is wrong. It could very well be, like you said, that the cytoplasm is so variable that both groups are right. And how do you actually create something meaningful out of that?
And if you want to address these problems technologically in the future, particularly gene expression problems, things that are ultimately regulated at such complex degrees, you're going to need to regulate the pattern rather than the actual instances. You can turn on and off this gene and that gene, but if there's a whole concert, like a whole orchestra of occurrences, including the shape change of the DNA, well, you need to really be on the lookout for sculpting these greater, broader patterns. And yeah, I'm really curious what you think the future will look like in terms of putting all these pieces of information in one place.
Carnage.
Carnage.
Absolute carnage.
Mayhem.
No, just mayhem. Absolutely. I do think that we're getting towards like looking at things. There's kind of the top-down approach and the bottom-up approach, and sort of going from what you know and then trying to make it more complicated in a biological context. But then there's also then taking all of this data and then trying to decipher something meaningful out of it. And I do think that computational abilities are really moving forwards. There's a lot of people using machine learning to try and solve some of these problems and find patterns that we can't actually see or visualize, but a computer, once they figured it all out, can. So I think that's going to be really interesting moving forward. A lot more availability of data now. So people used to do lots of big experiments but kind of keep the data to themselves. But there's a big movement towards being more open about data and it being available for others to use. And the availability of all of that data is sometimes quite overwhelming. Where do you go? And in particular for work on the human genome. So in the UK, we have a project run by Genomics England where they initially wanted to do 100,000 to determine the sequence for 100,000 people, that their genomes, so the 100,000 Genomes Project, and they're going forwards with that. And our National Health Service was going to be taking or getting the genomic information from all newborns. COVID's kind of put some of these ideas kind of off the rails for a little bit, but actually the real potential of being able to kind of using that anonymized data to work out, actually, well, this person has this illness and this person has this illness. They don't know that it's kind of related to this other thing that connects them. Being able to make those connections in that data is going to be really, really exciting moving forwards. So I do think the future is going to be a lot more computational. It's going to involve a lot of information that perhaps people might be feeling uncomfortable with others, knowing. I think there's people have some real worries about, well, if they know my genome, well, what else are they going to do with that data? But if that data remains anonymized, then I think we can learn a whole lot from it. And it's ripe for the picking. And
it brings up this other idea that there's almost a new class of scientists emerging which are analyzing the other scientists' work. Like there's this meta science which is becoming more and more important as the data science improves, and this is kind of a dream come true in a sense, because it's always the dream to remove the biases from the scientists who are doing their work so that they're not interpreting it just so they can get a new grant or something. And so it's really cool that you have these data scientists coming forth who are digging through other people's research and trying to pull patterns out and see what's happening and what's not working methodologically. And I think this is a really good sign, actually.
But the question is, does it capture things that we don't know to look for yet, right? Because you say that perhaps B form DNA is not the default helix. You suggest that perhaps the way that transcription factors bind is not the way that we think that they bind. And does having these reams and reams and reams of data drive you towards reimagining paradigms, or is there a significant danger of allowing the sort of the tidal wave of information to overwhelm a theoretical turnover? Because to my mind,
concept.
It is a really big concept, but like to my mind, I imagine that I worry about this sometimes, where you basically have a situation where you collect data and you analyze it according to a model that has been established. And the model is very, very functional, and it allows you to sort of to make a lot of conclusions, and it allows you to draw parallels, and it allows you to create a map. But in reality, it's functional despite the fact that it hides a fundamental error in the model itself. Does that make sense?
Don't you think that meta-analyses would deal with that to some extent?
I don't know. There's a really good meme about meta-analyses, which is it's one of those, like, on the left side, it's there's this guy who looks like he's in front of like an airplane window, and it's like so the meta-analysis is him like in front of the airplane window, and then it zooms out and it's actually him holding up like a toilet seat cover in front of like a picture of an airplane, and it's like that's the papers that are actually inside the meta-analysis. And I think you can have a lot of junk that goes into a meta-analysis, and it doesn't necessarily solve the question of how good is the information that you're looking at, because there's this tendency for us to be like, sufficient quantities of information will lead us to the right answer.
You do have the opportunity though to see data, like to see studies that are outliers, right? Or these studies contradict these studies.
You do, and
maybe this method is less reliable, or this group is less reliable.
Yeah, I guess I worry that a significant quantity of sequencing information could obscure the importance of discovering ways in which DNA works that aren't the conventional way.
Yeah, I mean, I guess you could see it that way, but I don't think we're ever going to get people just working on one area. You're always going to get a mixture of people. Like at the moment, I'm not doing anything in that area. I'm kind of interested in the bits that I'm doing. And you will always have that in science. You're never going to get everybody just doing one thing. And I think that's good. I think that's good that people do different things, that they're interested in different things. I sometimes think that, funding could be a little bit more diverse in what it wants to, what we're funding, rather than kind of forcing people to kind of chase pots of money. Blue sky thinking's really required in science. And trying to think about things different. Sorry?
I said I've never heard the term blue sky thinking.
Blue sky thinking. It's like thinking of so you would use that term for doing science for curiosity's sake, rather than kind of looking for a particular application. Mm-hmm. It's like, so why is the sky blue? Like, some people might say, well, it's not going to change colours, what's the point? Carry on, or do something, do some application.
I think those are engineers.
Probably. but yeah, so curiosity driven science, I guess, is probably a more accurate way of describing it, and actually thinking about things and trying to think outside the box rather than be kind of swept along with the latest craze.
I guess I think that strikes exactly at what I was trying to say, is that the sort of the sequencing and these like huge agglomerations of genomes seem like they're projects that draw a lot of money. And they do seem a little bit like a craze where people are like, we'll unlock something very fundamental by being able to aggregate all of this data. But I suppose my fear is the fact that it would, because I wish that science wasn't a zero-sum game, but it often does feel like a zero-sum game, where I'm like, look, there's a limited pot of money. And when you're like, look, shiny, flashy computer sequencing, I imagine that there's a risk that it would draw that pool of money in that direction, rather than allowing it to remain in the hands of the blue sky research, right?
Yeah, so I mean, blue sky research is really is really about kind of thinking of new ideas and thinking of, why is this happening? Rather than kind of looking at applications, just looking at trying to kind of broaden the edge, thinking about things differently. But then I think sometimes funders are really kind of thinking about what's the application of that and what can we do with it. But actually, sometimes the application of an idea comes 50 years later, or several very creative scientific concepts suddenly become a new way to do something that's really important. I think trying to kind of connect science with an immediate application can sometimes be beneficial, but also can sometimes actually stop us from looking at the new things.
Mm-hmm.
Absolutely.
And
it also sort of confuses people about what science is, right? People we love to talk to people on all sorts of cutting edges, especially this comes up in physics all the time, where people will be be like, well, we don't really understand like what causes this phenomenon. They'll be like, oh, well, we have, GPS, or we have all these technologies. And it's like, well, the ability to instrument, like electric current's a great example. We just did an episode on electric current. And, obviously, since we're talking from thousands of miles away to each other, we're very good as human beings at manipulating electricity. But there's a great deal of confusion as to what is actually happening inside of a wire, despite how, physically, what is the picture of what's happening? What is the scientific mechanism of what's happening? It's not so clear, actually. And so there's this huge conflation of science with technology in the general public, and really among a lot of scientists as well. And it's funny because you'll see a research institute and it'll be like the blank and blank research institute of science and technology, right? People do on some part of their brain recognize that science and technology are separate things. But at the end of the day, when you're applying for money, it's like, everyone's like, well, tell us what we can do. What can we make out of this science, So it's kind of it's a confusion. It's a cultural confusion in a way, I think.
Yeah. Do you feel the pressure in your own work to sort of to play to that?
That's kind of an interesting one. I think because there's almost a direct application of things that I do that it's not something that I've been concerned about. There was right at the start of my career, there was this whole motifs don't exist. They only form at acidic pH, so they're not physiologically relevant type thing that I had kind of had to argue against. But I guess that's not relating to application. That was more relating to don't believe that these things exist type thing, which is more of the issue that I've had in my career so far.
So you've been fortunate enough that people care about the science enough to fund you for this.
Yeah, I still, despite some people are like, these things don't exist, so let's not bother. But not everybody's like that. Some people have the open mind, and actually, I think we should all have the open mind. That's why I find the difficulty with persuading people or persuading funders to give you money. Like sometimes I think, well, actually, what I think I'm doing is really interesting, but the next person along there, I might not think it's interesting. They might not think it's interesting, but actually that could be the next life-changing medicine or the next thing, but we just don't know it yet. And we don't know what we don't know. Mm-hmm. And that's the problem. And
that's why we need explorers, right? That's why it is actually worth funding exploration. Because
so funding creative, new creative ideas or new scientific concepts, or looking for things that we don't yet fully understand, or we can't plan what the outcomes are going to be. That type of curiosity driven blue sky research I think is really important, but also it's very difficult to get funded because funders want to know, well, is it going to work? Mm-hmm. Otherwise, this might be a waste of money. Like, well, it might. I don't know. It's high risk, high reward, I guess.
Yeah. Is there a discipline that you look at where you think that it will be very important for the future that's not getting that hasn't yet gotten the attention that it deserves?
Oh, that's a difficult question.
Yeah, I don't mean to drop you that on the top of your head like that out of the blue. I guess I often wonder where it's like, we're entering into a phase of bioinformatics and sequencing and these sort of big computer. We're entering into the computer era of science, which is going to uncover a lot of these connections, like you were saying, that we don't necessarily even have the ability to know are there right at this moment, simply because we don't have the volume of genomes that are necessary for creating these correlations. And so, does that mean that sort of, if people are looking to go into science or looking to understand science or looking to participate in science, should they be veering in the direction of statistics and bioinformatics and programming? Or is there still the sort of the hard wet lab science of bench work that still has a huge, that still has a revolution ahead inside of it? Because it seems like this question of how do we look inside of cells has not yet been answered. And so my question is, do you think that we will head in the direction of figuring out tighter and tighter ways to look inside of cells? Or will the focus start to shift more towards, well, let's analyze what we have because we have this huge backlog of information that we need to process to get something meaningful out of it?
I don't think data scientists are going through, there's a massive backlog, we need to get through this. I think that kind of, there is a massive backlog of things that you can do. And I think there's great potential for people that are interested in informatics. I do think moving forward that if I was training now, I would really want to be doing more bioinformatics, because I do see it as the future, even if you're doing wet lab. And I think they're going to go hand in hand, and the next generation of scientists, it's going to be a hygiene factor in their training, that they're not just going to do stats, or if they're a biologist, for example, they're also going to do sort of learning how to program and learning how to process these large amounts of data, and it's just going to be second nature to them. I think that's kind of the way things are going moving forward. But then techniques that make things easy just sort of revolutionize what people do in the labs day to day. Something that might have taken somebody who is working alongside me in the lab months, you can just send it off and get the answer a day later, like for cheap. And that's just always going to happen. We're always going to have new methods to make things a bit easier. I don't think that's going to stop people doing wet lab experiments. I think they're essential, and I think people are interested in them. And if people are interested in them, then you'll have people working on them through some way or another.
I like that.
So I wanted to ask you, we've been talking thus far exclusively about these alternative structures in humans, but I noticed that you had also done some work, was it in agriculture, right? That had sort of, not in agriculture, but that had some interesting implications perhaps for agriculture, particularly with was it the nitrate utilization and the excess bacterial growth in the soil? And can you talk about that a little bit? It was quite fascinating, because one thing I didn't realize is that this nitrous oxide was a greenhouse gas, and it was actually something like 300 times more radiative potential for trapping heat than carbon dioxide, which is generally thought of as our big baddie in the room. Can you speak to that briefly? It's quite a fascinating story from what I can tell.
Yeah, sure. So this is work that I do in collaboration with a fantastic scientist at the University of East Anglia called Dr. Andrew Gates. And we work in collaboration with Professor David Richardson on this. So they are focused on the microbiology and interested in the bacteria, the soil bacteria, and also how they respire and how they grow and how that affects the soil, but also how it affects the atmosphere. And I'm obviously interested in DNA structures. And I met Andy at an event where he obviously knew that his bacteria were doing these weird things. And I was talking about my weird DNA. And he was just like, I think there might be a connection here, which is kind of cool. So the bacteria that we're looking at is a model soil bacterium called Paracoccus denitrificans. And this bacteria can grow on ammonium, and it uses ammonium to make its amino acids and its all of its biological materials. It's a source of nitrogen, but it can also grow on nitrate, which is obviously a fertilizer. And we are using fertilizers all across the world, and actually using those fertilizers affects the way those bacteria grow. It affects the way that they respire. And we're looking at the relationship between the different types of nitrogen that they're able to grow on, how they process that, and then what the output is. So in some ways, they can make nitrous oxide. And as you pointed out, this is a global warming gas. It contributes, I think, about 17% of full global warming emissions, which is quite a big lot for something that most people have not heard of. They're like, oh, we've had a carbon dioxide, we know that's bad. We also know that methane's bad because, cows and things like that.
And this is a topic that comes up on the show all the time, is how humans are fixated when it comes to environmentalism. It's like, well, we got one thing, it's called carbon dioxide, and if we just deal with that, everything will be fine. And it's an unfortunate myth that I think persists, especially in political realms, but even among scientists, where it's like there's just a litany of chemicals that are very concerning, to say the least. And I was shocked. I haven't heard about nitrous oxide. So please continue.
Yeah. So I mean, we were looking at we found a quadruplex in front of some genes that enabled that bacteria to grow on nitrate. So the bacteria can grow on ammonium, and it just takes the ammonium in, uses it. But if it wants to use the nitrate, it has to convert it to ammonium. And then to do that, it uses some proteins. And I think in the paper that you've probably found, those proteins were in the NAS operon. So we found that there was a quadruplex nicely positioned to control that, the expression of those proteins. And we showed that by adding compounds that we know stabilize that quadruplex, we can stop the expression of those proteins, and then be able to stop the bacteria from growing on nitrate. And then we could compare that actually. So we grew them in nitrate and we grew them in ammonium, and we were able to see the quadruplex specific effect because the ammonium growth was unaffected by these compounds. So we're funded at the moment by the BBSRC to look at other genes relating to this process. So that's something we're looking at in more detail.
Are you worried about those compounds affecting other things in the soil, like the plants themselves, or do you think it's fairly specific?
We haven't thrown them on any bits of soil so far.
Fair enough.
What we think at the moment, we're kind of trying to understand how these processes work. And it might be that there are some native compounds or natural compounds that we could use, or some other triggers that we could use that we could explore. But we, at the moment, again, trying to understand how it works. And once you understand how something works, then you can then think, actually, if we want to stop this from happening, what's the best way we can do it?
Mm-hmm.
Well, in some sense, bacteria are a fantastic effector system, because you're talking about molecular regulation, and what better place, you see an effect and you think, all right, these bacteria are doing some behavior, a very simple behavior that is sort of maybe not desirable in this farm field. It seems like your intuition is, well, this is such a common regulatory mechanism, we don't know much about it, it's probably happening there. It really just throws open the door to where else in the world this has extraordinarily important implications that we haven't even begun to uncover. So I'm really fascinated to see what comes of this as the years wind on.
Thank you. Well, we are obviously interested. Otherwise, wouldn't be here. But I do think there are a lot of other bacteria, in particular, I think will probably have some fascinating uses for DNA secondary structures, because obviously bacteria have a very small amount of non-coding DNA. So if there is a structure there, it's probably going to be functional, because otherwise the bacteria would have got rid of it a long time ago, because the more obstructions there are in their DNA, the slower it is going to them to divide. And obviously there's this sort of selection pressure that the quicker bacteria can divide, the evolutionary advantages that they're going to have, because the quicker you divide, the more mutations you're going to get, the better you can become, essentially.
Well, the annoying thing is that it seems like the more that you molecularly target fast-growing systems like cancer or bacteria, the quicker they're going to evolve solutions to that problem. And you kind of fight this war of this just ongoing feedback loop. But it'll be interesting to see if that can be just another tool in the toolkit, just like in situations where antibiotics become useless due to this same sort of evolutionary procedure. Maybe targeting some of these structures will provide a nice backup in medicine. Who knows?
Well, we'll see. It's certainly something that I'm interested in, and a lot of other people are interested in. And it might be that actually it doesn't come to anything, but I don't think that that's likely, because I think there's a lot of potential for lots of different opportunities that these structures can bring.
Cool. Well, I think it's really interesting stuff. I am very much looking forward to seeing how this plays out in the biomedical world, agriculture, and just from a scientific level. It really just opens up the sense of wonder for how unendingly complex these entities that we are really can be down on a physical level. And thank you for sharing all of this with us today. I really appreciate learning these things.
Yeah.
Thank you so much for having me and having the opportunity to talk science. It's good.
Yeah, and I just want to add that you have a YouTube channel, and you do this really wonderful thing that I wish all scientists would do, which is you try to prepare a very short 5-minute presentation for some of your papers as they come out in layman's terms, which I think is super valuable. I hope that you spread the word so that all scientists will take a page from your playbook there and get with it. That's a really wonderful thing in terms of motivating the next generation of researchers, and even allowing people who are, tangentially interested to familiarize themselves with your work. So I really applaud your effort there, and everybody should go out and check out your YouTube channel. Is there somewhere else people can find you? Do you have a lab website?
What's the name of the YouTube channel?
Oh, right. I think
I'm at Dr. Zoe Waller. It's the same on Twitter. I just try to keep it consistent. I do have I just have my departmental website based on UCL. I don't have my own personal research website yet. I just with everything I do so many other things, like running a website, a personal website as well on top of everything else.
Fair enough.
Can't do everything.
Indeed.
No, you can. All right, Dr. Waller, it's been wonderful. Thank you so much.
Thank you so much.
We'll hope to catch up with you later down the line.
Thank you very much for having me, and take care.
You as well. Bye.
Bye.